Welcome back. We're going to move into the respiratory system now. If you think about the respiratory system, it kind of bookends your life. You've got your kind of first cry, your first breath at birth, all the way to kind of your dying gasp. The driving force behind all of this breathing is the ability to make ATP. We make ATP most efficiently in the presence of oxygen, so the job of the respiratory system is going to be to supply our body with that oxygen so we can make ATP. It's also going to get rid of a portion of our carbon dioxide as a waste product. We actually have three different processes that are sort of collectively under the umbrella of respiration. We start with just the concept of the movement of air in and out. That's pulmonary ventilation. Gas exchange is then going to be the exchange of the oxygen and carbon dioxide between the blood and whatever it is we're talking about. External respiration is gas exchange in the lungs. So this is where the blood is dropping off carbon dioxide and picking up oxygen. That blood then travels down to the tissues where internal respiration occurs, and we're going to have the gas exchange there where we're going to drop off the oxygen and pick up the CO2. The cells then use the oxygen via cellular respiration to make the ATP. So we have multiple different processes here. We divide this system into two zones or divisions based on whether or not gas exchange is occurring or not. So anything that's going to be found in the conducting zone, there's no gas exchange occurring. This is just tubing that gets the air to the respiratory zone where gas exchange can occur. So the anatomy of the system is fairly straightforward. We've got the nose, the sinuses, the pharynx, the larynx, the trachea, the bronchial tree, and then into the lungs where we eventually end up out in the alveoli. The conducting zone, where there's no gas exchange, basically goes from the opening of the nose to your nostrils all the way into the bronchial tree to a level called the terminal bronchioles. It's fairly deep into the system that is simply conducting. The respiratory zone then picks up there in what are called respiratory bronchioles and goes all the way to the end, which would end in the alveoli. Clinically, we also tend to divide this into an upper respiratory tract and a lower respiratory tract. This is useful in dealing with infections and whether or not we're dealing with something that is more upper or lower. So we'll start at the beginning with the nose. The nose is designed to function solely for breathing. And so when we breathe through our nose, we're actually going to process the air. We're going to warm it up to body temperature. We're going to filter it through the kind of sticky, mucus-covered nose hairs, and we're going to add some humidity in. When we breathe through our mouth, we don't get these benefits. So when we go and we take a big, deep breath through our mouth on a very cold day that's generally fairly dry, we tend to cough as a reflex because we don't have the surface area in the mouth to properly warm up the air and humidify the air. Obviously, our sense of smell is also going to be located here, and it is going to help amplify our voice out as well. That's why your voice sounds different when your nose is all stuffed up. So this is going to extend basically from the nostrils, or what are often called the nares, all the way to the posterior nasal apertures, which is the opening into the pharynx. We basically have a bunch of hyaline cartilage and bone that is going to shape most of this. And it begins with kind of what's called the tip of your nose, or what's called the vestibule. There is a significant number of hairs called the vibrissae that are guarding this area. So most of your nasal hairs are right here at the very beginning in the vestibule, as opposed to way back in the cavity. Now, the surface area is increased by these projections called the nasal concha that project out into the cavity, And then the meatuses are kind of the valleys between them. And so that's going to create extra surface area that is going to help slow air down as it has to work through these kind of caverns in the nasal cavity. And that's going to give us the surface area and the time needed to warm up and humidify the air. Now, usually we're going to be talking about respiratory epithelium for the majority of this system. So remember that is pseudostratified ciliated columnar epithelium, lots of goblet cells, really nice cilia to help move all that mucus. The very, very top, the superior portion, way up by the olfactory bulb, is going to have olfactory epithelium that deals with your sense of smell, but the majority is respiratory. And then the nose is incredibly vascular. That is why it's going to bleed fairly easy without a huge amount of pressure put on it, because you have a huge capillary network in there. So here we can kind of see the cavity here. So these are your concha, and then the meatuses are the spaces between them. Here you can see that very clearly on the cadaver. So this is your nostril where we start. This is that vestibule where you're going to fill with your nose hairs in here. And then we go back to the posterior nasal aperture that's going to then lead us into the pharynx. So the pharynx is basically a muscular funnel. It runs between the nasal cavity all the way down to the larynx and is divided into 3 regions. The most superior is called the nasopharynx. So here it's indicated in yellow, and it is shown on the cadaver right here. This runs basically from that posterior nasal aperture down to the level of the soft palate, which is this structure right here. Couple different things going on here. First of all, the pharyngotympanic tubes from our middle ear open into the nasopharynx, and we have a pharyngeal tonsil back here that is often more commonly known as the adenoids. There's a full 90-degree turn. So if you look here, you go here, down. So there's a full 90-degree turn here. So that's going to mean that really anything that would get moved into the nasal cavity that would be fairly large would not really be able to make this very narrow 90-degree turn, and that is going to be protective. So we're going to come down to basically the level of the uvula. So the uvula is the most distal part of the soft palate. So this is the hard palate here, and then the soft palate is all of this. The most distal end is the uvula. So when you get to that, you know that you are basically going to be transitioning into the next portion of your pharynx. This area of the pharynx is strictly respiratory and is going to therefore be lined with respiratory epithelium. Continuing on from there, we get to the oropharynx. So we run between basically that uvula down to the level of this cartilage called the epiglottis. It is posterior to the oral cavity. You can see right here, this is your tongue, and it's shown in pink over here. Here we have the palatine tonsils that are much bigger. Because the oropharynx and what comes next, the laryngopharynx, are shared spaces with the digestive system, we actually see a change in epithelium. We're going to go to stratified squamous in these areas because everything you swallow is going to move through this area, and swallowing is abrasive and is going to require more protective type of epithelium. The most distal is going to be the laryngopharynx that runs from the epiglottis down to the level of the cricoid cartilage here in the larynx, so you can see right here. It then feeds directly into the esophagus. The laryngopharynx is where we're then going to split the respiratory system going anterior and the digestive system running posterior. Again, lined with that protective epithelium. That's going to lead us into the larynx, which is more commonly known as your voice box. The primary function of the larynx is actually to maintain a patent airway, so maintain an open airway, and make sure that anything you swallow doesn't end up in the airway. So the primary piece that is going to really work on this concept of making sure that we don't swallow into the respiratory system, is a piece of cartilage called the epiglottis. And we're going to see that it's going to fold down over the top of the larynx when we swallow. The fact that we have evolved the ability to make sound with this is a secondary role. It is not its primary role. The primary role is actually to make sure that you, A, always have an open airway, but B, that you're closing it off as needed when you're swallowing. The larynx is made up of 9 different cartilages. 3 are very large and they're singular, meaning there's only one of them. So you've got an epiglottis, a thyroid cartilage, and the cricoid cartilage. Then you have 3 small paired ones. So you have 2 arytenoids, 2 corniculates, and 2 cuneiforms to give you your 9. All of these except the epiglottis are hyaline cartilage. The epiglottis, of course, is going to be elastic cartilage. So here we can kind of take a look at this concept of the larynx. So the hyoid bone sits right here, and here you are seeing the 3 singular cartilages. So this is the epiglottis, the thyroid, and then the cricoid. So this is what you're looking at here. Now the thyroid cartilage does come to a point at its anteriormost point called the laryngeal prominence, or what is more commonly known as the Adam's apple. So the Adam's apple is absolutely not specific to Adams. There are more prominent laryngeal prominences, generally speaking, in males, which is how it got its name. That has to do with the fact that there are a significant number of testosterone receptors on the larynx, and during puberty, this whole structure is going to have a growth spurt in a male. It grows much slower in the female, and so we don't tend to see this massive growth spurt that creates huge changes in the voice like we do in the male. So when that happens, it tends to make this point very prominent. That being said, it can be more prominent in a female. You could have a male that doesn't have a significantly prominent thyroid cartilage. So it is no bearing on the actual sex. It is simply how large the thyroid cartilage is. Here we're looking posterior. So here you can see the epiglottis, the thyroid. You can see that the thyroid cartilage does not go all the way around. It stops. And then the cricoid, this is what it looks like on the anterior, and then on posterior it tends to get a little bit bigger. Looking at a sagittal view, so this is your thyroid here, this is the cricoid, and then this is the epiglottis. You can picture when you swallow, so the esophagus is right back here, this is going to flip down and it's going to cover up this opening to your trachea. So this elastic cartilage is going to flip down while you're swallowing and then pop back up. Now, the 3 paired ones are very small, so here's your arytenoid, there's your corniculate, and then embedded in the wall are the cuneiforms. We also are going to have these 2 folds that are going to extend inward in the larynx. We've got the vestibular folds and the vocal folds, or what are commonly known as the vocal cords. So the superior ones are the vestibular folds, the inferior ones are the vocal cords. So here looking at the larynx, the superior would be vestibular, the inferior would be vocal. Now, the vestibular play no role in speech. They are simply there to close the larynx off while you're swallowing. Even after the epiglottis has flipped down, we also close the airway. The inferior cords produce sound as air passes up between them, and they vibrate. Because they vibrate, they actually have a different type of epithelium. They're going to have our stratified squamous, even though we are going to have respiratory epithelium all around it. The vocal cords are going to be stratified squamous. The glottis is the vocal cords and then the opening between them. So the reason that this cartilage is called the epiglottis is that it's above the glottis. So here we are actually looking down. Here's your epiglottis here. And so here are your vocal cords, and then this is the opening into the trachea between them. So this structure right here is called the glottis. These are the vestibular folds up here that will help close this off while you're swallowing, and then the epiglottis would flip posteriorly to cover it up as well. So this should be telling you that we have a lot of different mechanisms to make sure that what we swallow ends up in the esophagus and not in the respiratory system. So we have muscles that are attached to the vocal cords that are going to allow us to either AB or adduct these cords. As you force air between adducted cords, it's going to cause them to vibrate. If the cords are very taut, it's going to create a very high-pitched sound, think like a child's voice. And if they're more slack, they got a little looseness to them, it's going to be lower pitched. Generally speaking, adult male vocal cords produce the lower pitches of the sounds. They're longer, they're thicker, they're more slack, they vibrate slower, and you get a lower sound. Loudness is determined solely by the amount of force that you're putting into pushing the air up. It doesn't have to do with any anatomy of the cords. Now, the vocal cords simply produce a buzzing sound. You have to shape that into recognizable sound. So here we can kind of get this idea of AB and adducting the cords. So it is the arytenoid and corniculate cartilages that are attached to these muscles that allow you to either close it off fairly well or open it up more. So our larynx leads into the trachea, which is the windpipe. It basically is going to run fairly straight down the mediastinum for, a few centimeters. It sits just anterior to the esophagus. And if you're thinking about, the windpipe, we think, this needs to be open in order for us to get our oxygen. So to help support and reinforce it, it's going to be made up of these C-shaped rings of hyaline cartilage. Remember, hyaline cartilage has a lot of tensile strength to it. So even if you try to compress the trachea, it's very difficult to do that because the hyaline cartilage maintains a patent opening. It also keeps it from collapsing from the inside when you are inhaling and exhaling and changing the different pressures that are occurring. Now, a C-shaped ring is not a complete ring, which means there's an opening. That opening faces posterior toward the esophagus. And that opening is spanned by a muscle called the trachealis muscle. The gap actually allows you to swallow. So the esophagus is essentially a potential space, not an actual space. It is closed down completely unless you are actively swallowing something. In front of it is the trachea. Behind it is the body of the vertebrae. And so it's really stuck between a rock and a hard place here. When you go to swallow something, it has to expand in one direction or the other. It cannot expand posteriorly because the vertebrae are not going to move. The trachealis muscle is going to give the esophagus something soft to expand into. If you take too big of a bite and you swallow too much at one time, sometimes it hurts. You can kind of feel it working its way down there. That's because it's pushing on the cartilage. So you've gone past the point where the trachealis muscle is, and now you're actually pushing on the cartilage, and it hurts. This muscle will also respond to sympathetic nervous responses and will relax to allow more airflow to occur. Okay, so the inner lining of the trachea is going to be that respiratory epithelium. So we've got a lot of cilia here and a lot of mucus. This is going to combine to create what's called the mucociliary escalator. All of that cilia is constantly beating upward, and the mucus is traveling with it. So all of the debris and pathogens and anything that comes in with what we breathe is constantly being beaten upward. And usually you're not producing enough of it that you notice it, you simply swallow it. If you're sick or something like that, then you tend to produce more mucus and it tends to be more noticeable. The middle layer is where we're going to find more glands, so mucus glands and serous glands, and then our lymphatic system. This is where we find our MALT, so that mucosal-associated lymphatic tissue we talked about. The respiratory system is a huge portal of entry for pathogens because we don't have a lot of specificity in what we're breathing. We just basically breathe whatever's there. So we definitely want to have the immune system right beneath the surface, ready to take care of whatever the issue is. The outer layer is called the adventitia. You've got this fibrous connective tissue that kind of blends into the other organs in the mediastinum. You also have the cartilage rings here, and eventually you get all the way down to the branch point where we're going to find a piece of cartilage called the carina. So here are our C-shaped cartilage rings. This is what they look like. Here's that trachealis muscle. So you can picture the esophagus right here. And so when you swallow, the esophagus expands anteriorly into this muscle, and that allows you to swallow there. So here's the concept of the mucociliary escalator. There's a constant movement upward. This is what the cilia look like. These are your goblet cells that are consistently releasing mucus. And so you can imagine that if all of this begins to move in one direction, it's going to be like kind of like seaweed. Okay. So we're going to come all the way down. So this is the cricoid, and beneath that we start the tracheal cartilages. And then we get to this piece called the carina, which is going to be a little bit more than just a ring. It's going to have some cartilage that actually moves down into the branch point to support this point right here. This is essentially the weakest point of the trachea because it's where it branches. And so we've got some extra cartilage here to provide that support. The trachea then is going to lead us into the bronchial tree. And so you can see the beginnings of it here. And really we're going to be building this concept of like a tree. You want to picture the trachea, just take this whole thing, flip it upside down, and put the trachea in the ground. It's the trunk of the tree. And now we're going to move into all of the branches of it. So you're going to basically start from your two main bronchi, and you're going to end up with 65,000 terminal bronchioles in each lung. So you start with two branches, and then you end up with 130,000 of these terminal bronchioles. And if you remember, the terminal bronchiole is the end of the conducting division. So this is a big tree. We haven't even gotten to where we're dealing with gas exchange yet. All right, so the main or primary bronchi are essentially extensions of the trachea, same C-shaped hyaline cartilage rings. The lungs are not symmetrical, and neither is the tree. So on the right main bronchus, we've got a short tube. So it's only about 2 to 3 centimeters long, and it sits more vertically and is wider than the left. So path of least resistance is if you aspirate something down into the trachea, it's going to go right most of the time. The left is going to be longer, narrower, and sitting more horizontal, so it would take more energy for something to move down to the left side. Doesn't mean it can't happen, but more often than not, aspirated things are going to end up in the right. So here's where we are. We're trachea, and then here are our two main bronchi. Okay. The main bronchi are going to branch into what are called lobar or secondary. We can see them here, here, and here. And then there's one here and one here. So lobar means lobe. The right has 3 lobes, the left has 2, so we have a grand total of 5 lobar bronchi. When we move from the primary to secondary, the hyaline cartilage is going to thin out a little bit. So instead of being a C shape, it's going to be more crescent shaped. So the edges are going to be thinning out. That same concept moves into the next branch, which are called the tertiary or segmental bronchi. Usually we have about 10 of these on the right and anywhere from 8 to 10 on the left. Each of these is going to go to what's called a bronchopulmonary segment. So if you look at this cast here, each color represents a bronchopulmonary segment. So each one of these colored segments would be getting a segmental bronchi. Now, if we look at the bronchi, we're going to be seeing respiratory epithelium. However, it's going to change as we go. The farther into the tree we get, the cells actually become shorter and shorter and shorter. And then eventually, by the time we get out to what we're going to call the leaves of our tree, the alveoli, we end up with simple squamous epithelium. So this is a place in the body where you have a very gradual transition. The cartilage changes very gradually, and the cells change very gradually in the epithelium. We have a really nice layer of smooth muscle in here. It's going to help contract and relax the airway. And we have a huge, huge amount of elastic connective tissue. We're going to see that this is going to be incredibly important in the function of the lungs. The elasticity of the lungs is absolutely imperative for the mechanics of breathing. It's going to contribute to the recoil that's going to help us push air back out. So the entire tree and then the surrounding tissue of the lungs is incredibly elastic. Again, like I was talking about before, the cartilage is going to change. It's going to thin out on the edges, so it's going to become crescent shape and then get really thin crescent, and then finally it's going to become fairly irregular, and then it disappears altogether as we get down into the bronchioles. So the bronchioles are where the cartilage disappears. We're talking about tubes that are about 1 millimeter or less in diameter. The epithelium has gone from a columnar down to a ciliated cuboidal, and we got a really nice layer of smooth muscle. The bronchioles then feed into terminal bronchioles. So each bronchiole is going to branch into anywhere between 50 and 80 terminal bronchioles, which are the final portion of the conducting division. We're talking about 0.5 millimeter or less. If you look at this histologically, mucus glands and goblet cells disappear because we're getting close to the respiratory division where mucus is going to hinder gas exchange. So we don't want there to be mucus down at the distal end of our tree. So these glands disappear. However, we still see cilia because the mucus drains down from above, and we need to, again, mucociliary escalator, beat everything upward. Each of these, so you have about 65,000 terminal bronchioles per lung, and each of those will give off 2 or more what are called respiratory bronchioles. So the respiratory bronchioles have alveoli budding out from their walls, and so they are considered the beginning of the respiratory division because gas exchange can begin to occur. So here you've got a bronchiole, this is a terminal bronchiole, and then this one's branching into 1, 2, 3 different respiratory bronchioles. Your respiratory bronchioles can divide into anywhere between 2 to 10 alveolar ducts that lead into alveolar sacs, which basically kind of look like little clusters of grapes, where each of the grapes would represent one alveoli. Now you can see that the alveoli have very thin walls and are going to be kind of open. Now we also have a nice blood supply coming through here. So this is the pulmonary arteriole coming off of the pulmonary arteries, coming out of the right side of the heart, carrying that deoxygenated blood in. Each alveoli is surrounded by a capillary network. Oxygenated blood is then going to travel back out the pulmonary venules, which will eventually lead up to pulmonary veins, which will eventually dump into the left side of the heart. So here we can get another view of this. So again, here is a bronchiole to a terminal bronchiole to a respiratory bronchiole with some random alveoli popping off of the walls here. And so this is the beginning of the respiratory division. We can see actually here's the duct. Okay, and then this is the bronchiole, and you can see histologically there's more going on in the wall here than there is in a duct. So again, this sort of progressive thinning of everything that we see. This is kind of a good picture. I pulled it from my histology book, but it just shows you that as you go, how things change. So your epithelium starts at, the ciliated columnar, and it then is going to end up with sort of ciliated cuboidal all the way down to simple squamous. The goblet cells are very high until you get down to about the level of the terminal, and then they disappear. Ciliated cells go all the way down to somewhere in the respiratory bronchioles. Glands stop at that terminal bronchiole. The hyaline cartilage is going to thin out significantly and disappear by the time we get to the bronchioles. Smooth muscle is still fairly decent all the way through, but will disappear eventually. And then elastic, the concept of that elasticity almost all the way to the very end, because that's a hugely important concept in the function of the lungs. So I just kind of like this picture. It divides it into conducting versus the respiratory, and really gives you a visual for how things change as we go through the bronchial tree. Now, if we picture our tree, the alveoli are the leaves. So we have about 150 million per lung. So that's about 300 million leaves on our tree. We have about 70 meters squared of surface area for gas exchange, which is the equivalent of a few parking spaces sat next to each other. So that's pretty significant if you think about it in that very small space that are your lungs. But we also have to remember that blood is moving very fast. And as the red blood cells come into the lungs, they're not going to be there very long. When you're at rest, it gets about 3/4 of 1 second to get that oxygen loaded on. So we need the surface area to account for the fact that it's not in there very long. So we've got 3 different cell types. We start with what are called the squamous or the type 1 alveolar cells. These are your simple squamous epithelial cells. This is where we do our diffusion. This is going to be about 95% of the surface area that we see. The type 2, or the alveolar cells, or great alveolar cells, are roundish. They're kind of cuboidal. They make up the remaining 5% of the surface area. Their job is to take care of the type 1s. So they help repair them if they're damaged, and they're also going to secrete something called pulmonary surfactant that we're going to talk about in a little bit. Then we've got the alveolar macrophages, or what are called dust cells. These are most numerous of all of the cells. They're kind of like robot vacuums. They just wander around the lumen of the alveoli, and then there's pores that connect them into another alveoli, a neighboring one, like a doorway in your house. And so if you got one of those robot vacuums, you know that it just sort of wanders around and picks up whatever it finds. That's what these guys do. You're going to lose like about 100 million of these a day under normal circumstances, as they basically just sort of clean everything up, and then they take a ride up the mucociliary escalator. If you're doing something that would increase the amount of debris, you're working in a coal mine, or you're living in a terribly polluted city, or you're smoking 3 packs a day, something like that, then you would lose even more of these. So here, this is really nice views of all of this. So this is our simple squamous epithelium that's making up the walls here. And so you got really very, very thin tissue. These are your macrophages here, so you can see them throughout. That's your pulmonary artery, so really kind of a cool view of all of this. So we've got this capillary network surrounding each alveoli. So here's capillary and here's your alveoli. And when they're sitting there together, they're going to form something called the respiratory membrane. This is what we have to exchange across in terms of gas exchange. So it's made up of 2 very thin cells. We've got the type 1 squamous alveolar cells, so these guys, and then, so right here, okay, and then we've got the endothelial cells of the capillaries. So it is very, very thin, and they share a basement membrane between them, right here. It's very thin tissue, but anything that affects the respiratory membrane will affect gas exchange. So we're going to talk about a couple different things later on that negatively impact the respiratory membrane, which then negatively impacts gas exchange. So the alveoli get us into the lungs. The lungs are basically cone-shaped, elastic. They're kind of like a sponge almost. They sit in opposition to the heart. So if you remember the heart, the apex sat on the diaphragm, and then the base was where the great vessels came out. Flip that for the lungs. The base sits on the diaphragm, and the apex, or the point, comes up above your clavicle. So they're a lot taller than people think. They have a costal surface, so all the way anterior to posterior and around the lateral sides, are pushed into the rib cage. And then the mediastinal surface faces the mediastinum, and that's where we've got a hilum, which is going to be an opening in the serous membranes that allow everything to move in and out of the lungs. That's also known as the root. Now, similar to the heart, the lungs have two different vascular systems. We've got the pulmonary arteries and veins that are taking deoxygenated blood from the heart into the lungs to oxygenate, and then the veins bring it back to the heart. That is not blood that is going to be used by the lungs. The lungs are fed from the bronchial arteries that come off of the aorta. So this is very similar to when we were talking about the idea that the heart cannot use the blood that's in it. It has to use the oxygenated blood that comes out of the coronary arteries, which branch off of the aorta. All right, so here we can actually see the sort of sitting here of the lungs. We've got the hilum here. So this is where we've got the bronchial tree and the arteries and veins all coming in and out. Here you can see this concept of kind of the costal surfaces that are being in contact with this. And then this is just a really nice view of all of the sort of close-up tissue here. It really does kind of resemble a sponge. And all of these alveoli are your little air-filled spaces. So the lungs, like a sponge would be, are crowded by the organs around them. And so they're not symmetrical. The right lung tends to be shorter than the left because the liver is pushing up from below, but it has three lobes that are separated from each other. The left lung is taller because it's not being pushed up from below, but it's more narrow because the heart sits left of center. So there's a full what's called cardiac impression on the left lung that helps make room for the heart. The left only has two lobes. Last thing to talk about here in the anatomy is going to be the pleura. So we have the serous membrane surrounding the lungs. We've got the visceral and the parietal pleura. So we know that visceral is going to be what covers the lungs. Parietal is going to be the outer layer, which in this case is attached to the mediastinum, the inner surface of the rib cage, and the superior surface of the diaphragm. Between them is the potential space called the pleural cavity. There should not be an actual space between them. There should be a film of fluid, some protein-filled fluid in there called the pleural fluid that allows them to rub up against each other very easily with no friction. So similar to what we saw with the pericardial membranes in the heart. So functions of the pleura are going to be just that, to reduce friction, which all serous membranes do that. However, we are going to see that the pleura are also going to play a huge role in the actual function of the lungs. You can't breathe properly if your pleura are not there to help you create the pressure gradients that you need to create to allow air to flow in. So when we talk about the mechanics of breathing, we're going to talk about the role of the pleura here. Here you can actually see, these are really good pictures of this. So this is the visceral. You can see that it goes into all of the different fissures here. And then the parietal is attached out here. So here it is on the diaphragm and attached to the mediastinum and to the rib cage. So we're going to be coming back to this concept of the pleura in our next video.